Welcome to this explainer. Today, we're diving straight into the incredible biological engineering of the human respiratory system. Now, we aren't just going to look at this as a basic list of anatomical parts. No, we're framing this whole journey as an absolute evolutionary masterclass in optimizing fluid dynamics and thermodynamics. Because honestly, it's one of the most perfectly tuned machines in all of nature. To really appreciate just how flawlessly this system is built, we've got to rewind a bit. Think back to a pretty monumental year. 1661. That's when Italian biologist Marcello Malpighi used an early microscope to finally, definitively prove how our airways intimately connect to our blood supply. He actually observed the capillary network surrounding the alveoli and frog lungs, establishing the structural basis for gas exchange. And why does that matter for us today? Well, it was the fundamental discovery that proved our lungs aren't just, empty air sacs. They're a direct, intimate gateway to our entire bloodstream. So here's our roadmap for today. We're going top down, starting with the airways architecture, then the mechanics of breathing, how we measure lung capacity, gas exchange and transport, and finally, brain control and adaptations. Okay, Section 1, the airways architecture, plumbing the depths. Let's start with the physical pathways that actually bring the outside world directly into the core of our bodies. The air travels down what's essentially a brilliantly designed branching tree. It kicks off at the trachea, what we call Generation 0, which is supported by these rigid cartilage rings. From there, it splits into the primary bronchi, and then just keeps branching further and further into tiny cartilageless tubes called bronchioles. That covers generations 4 all the way through 15. This whole section, that's your conducting zone, and it's designed purely for transport. Now, the physical consequence of all this branching is absolutely mind-blowing. As the airways divide into millions of these microscopic tubes, the total combined cross-sectional area increases exponentially. And because of this massive increase in area, deep down in the alveoli, the forward velocity of bulk airflow completely stops. It literally drops to virtually zero. Down there in the respiratory zone, oxygen isn't blown around, it moves across the membrane purely by molecular diffusion. Moving on to Section 2, the mechanics of breathing, the physics of physiology. To understand how we actually pull air into that complex architecture, we have to look at pure classical physics. Breathing is entirely driven by pressure gradients, and it's governed fundamentally by Boyle's law. You probably remember this one. At a constant temperature, the pressure of a gas is inversely proportional to its volume. So what does that mean for us? Simply put, our body expands its thoracic volume to drop its internal pressure. That physically forces outside air to flow in, right down the pressure gradient. We don't actually suck air in. We create a vacuum, and atmospheric pressure does the rest of the heavy lifting. It's also really critical to distinguish between quiet and forced respiration. Go ahead and take a relaxed breath right now. Notice how exhaling takes zero effort? During quiet, resting breathing, inhaling is active. Your diaphragm is contracting, but exhaling is entirely passive. It just relies on the natural elastic recoil of your lungs springing back into place. Forced respiration, on the other hand, recruits a whole new team. It requires active muscle contraction from your internal intercostals and abdominal muscles to forcefully compress the chest and physically push that air out. Let's follow this biophysical cascade step by step for an active inspiration. Think of it like a chain reaction. First, a neural trigger fires, which flexes the diaphragm. That muscle contraction expands the thoracic cavity. As the cavity expands, the intrapleural vacuum pressure drops even further. That vacuum powerfully pulls the lungs outward, causing lung distension, which finally drops the alveolar pressure below atmospheric pressure, and boom, air rushes in. But there is a pretty massive biophysical problem hidden deep in the lungs, and it's defined by Laplace's law. Because there's a thin layer of water lining the alveoli, it creates surface tension. And Laplace's law tells us that a smaller alveolus actually has a higher collapsing pressure than a larger one. So if left completely unchecked, physics dictates that your smallest airways would literally crush themselves. Fortunately, there's a biological fix, and it's an absolute game changer. It's a specialized lipid-protein mixture called pulmonary surfactant. Without this mixture lowering that surface tension, those smaller alveoli would simply empty their air into the larger ones and completely collapse, a condition known as atelectasis. But thanks to type 2 alveolar cells dynamically secreting this surfactant, the collapsing pressure is stabilized across all the alveoli, no matter their size. Section 3, measuring lung capacity, the clinical spirogram. So, how do we actually measure the efficiency of this complex, pressure-driven mechanical pump? Well, we use a clinical spirogram to map our pulmonary capacity. It's actually a fascinating piece of medical history. This diagnostic technique was born way back in 1846. An English surgeon named John Hutchinson invented the water spirometer to evaluate the respiratory fitness of coal miners. He even coined the term vital capacity because he noticed how strictly this measurement correlated with the miner's overall likelihood of survival. We can rapidly break that spirogram down into 4 distinct standard lung volumes. You've got your normal tidal volume, your inspiratory reserve, your expiratory reserve, and, crucially, your residual volume. That residual volume, which is about 1,200 milliliters, is the air remaining in your lungs even after the absolute most forceful expiration you can muster. You literally cannot breathe this out. It acts as a mandatory buffer to keep your alveoli from fully collapsing. And notably, it's the one volume basic spirometry just cannot measure. All right, Section 4, Gas Exchange and Transport, the chemical handoff. Once that fresh air finally reaches the deep respiratory zone, our story shifts away from physical mechanics and steps right into some really beautiful biochemistry. The actual crossing of oxygen into the blood is governed by Fick's law of diffusion. This mathematical principle states that the rate of gas diffusion is proportional to the surface area and the pressure gradient, but inversely proportional to the membrane's thickness. And this functions flawlessly in humans because our respiratory membrane is astonishingly thin. We're talking an average of just 0.6 micrometers across 6 distinct anatomical layers. Once oxygen crosses that incredibly thin barrier, 98.5% of it binds to the transport protein hemoglobin. But hemoglobin needs to know exactly when and where to let go of that oxygen at the tissues. If you want an easy way to remember what local environmental changes force hemoglobin to release its oxygen, just memorize this clever mnemonic. Cadet, face right. That stands for high carbon dioxide, acid, 2,3-DPG, exercise, and elevated temperature. Now that right shift physically means hemoglobin lowers its binding affinity. This right here is a marvel of allosteric biochemistry. The hemoglobin molecule literally changes shape, forcing it to dump its precious oxygen cargo exactly where active, exercising, acidic, or hot tissues need it most. It's essentially this brilliant, autonomous local delivery system. Finally, Section 5, Brain Control and Adaptations, the autopilot software. To keep this entire system running continuously, the brainstem acts as an incredible emergency responder. In a respiratory compensation reflex loop, let's say your body suffers a primary defect, like metabolic acidosis. Your arterial pH drops to a dangerous level. Peripheral chemoreceptors in your arteries instantly detect this and stimulate the medullary respiratory centers. The immediate response? Hyperventilation. Your lungs kick into overdrive to literally blow off volatile carbon dioxide, which shifts your buffer equations and pulls your blood pH right back to normal in a matter of minutes. Which leaves us with a pretty fascinating final thought. If this respiratory masterclass can perfectly balance Boyle's law, Laplace's law, and Fick's law right now, while you are just sitting there resting, how far do you think evolution can push these exact same biological rules? I mean, consider marine mammals. They possess extreme adaptations, like total alveolar collapse, just to survive extreme, oxygen-starved environments under the crushing hydrostatic pressure of the deep ocean. It really just goes to show that biology is the ultimate engineer. Thanks for diving deep into the respiratory system with me on this explainer. Keep learning and keep marveling at the machinery inside you.